Blots. It is a molecular biology technique that checks for the presence of a specific sequence of nucleotides or protein in a sample. Southern blot. It is a method used to detect specific DNA sequence in a given DNA sample. DNA samples are separated via electrophoresis on a gel. The separated DNA is transferred to a filter. The DNA is exposed to a labeled DNA probe that anneals to its complement. The labeled probe is visualized, locating the DNA sample of interest. Northern blot. It is used to study gene expression by detection of RNA in a sample. It is the same as southern blot, except the sample is RNA and not DNA. The probe remains DNA. Western blot. It is used to detect specific proteins. It is similar to other types of blot, but protein-specific antibodies are used for detection. After the proteins are electrophoretically separated and transferred to a filter, a primary antibody is applied. The excess primary antibody is washed off and secondary antibody is applied. Secondary antibody recognizes and binds to the primary antibody. Finally, an appropriate substrate is added to visualize the bands of interest. The Western blot is the confirmatory test in a presumed HIV infection. Southwestern blot. It is used to study the interaction of DNA with DNA-binding proteins, such as proteins that regulate transcription. Proteins are separated on a gel via electrophoresis, then they are transferred to a filter. Proteins are exposed to radio-labeled known DNA sequences. The proteins that bind the sequence will bind the DNA, which can then be visualized on a film.